All right. Want to thank you very much for joining us. This is our monthly webinar hosted by the Florida Invasive Species Council's Partnership Assistance and Communications Committee. And so we're here to help address invasive species management across boundaries and help support our cooperative invasive species management areas, or CISMAs, that cover the entire state of Florida, working regionally on invasive species issues. So for today's presentation, we have Dr. Susan Wilde. She'll be presenting on, and hopefully I'll say this properly, the avian vacuolar myelinopathy update. And if you give me a second, I will go ahead and hand the controls of the presentation over to her while I introduce, give her introduction. Go ahead and stop my sharing. Now, as we go through the presentation, feel free to put any questions that you have in the chat. And if you can, keep yourselves muted during the presentation so we can focus on her information, and we'll address any questions and start sharing. Associate professor at the University of Georgia in the Warnell School. Well, previously employed as a research scientist within the South Carolina Department of Natural Resources Marine Research Institute. She was focused on merging management concerns and targeted research to optimize management in critical and immediate aquatic natural resource problems. Dr. Susan Wilde's research and teaching at UGA continues to focus on impaired aquatic systems and interdisciplinary challenges with invasive plants and animals, nutrient pollution, harmful algal blooms, emerging contaminants, and their implications for fish, wildlife, and even humans, depending on those valuable water resources. All right, and so go ahead and hand it over to you.
All right, thank you, Deb. Can y'all hear me now?
Yes.
Great. See, I came from Illinois, but I learned how to say y'all. It's a very useful word, more than you guys. All right, so today I'm going to talk about avian vacuolar myelinopathy, but now we refer to it as vacuolar myelinopathy because it's killing more than avian species. So we'll talk a little bit more about that today, and very interested in having some more help on the invasive species issues because I think we have kind of a meltdown in Southwest Florida concerning this toxin. If I can get it to move forward. So I have been working on this for a very long time. I personally started on it 25 years ago, but the initial eagle deaths occurred in Arkansas in 1994 and 1996, and there was an unexplained epizootic with lots of neurologically impaired eagles and coots in those locations in Arkansas. But we have moved on to do more extensive reservoir monitoring in all the locations where it now occurs, and a lot of field studies and food chain transfer trials. So now we know it's occurring in more locations. We have discovered the toxin, and we know there's more species that are affected by this toxin and develop vacuolar myelinopathy. So we now have concerns for aquatic mammals and aquatic people. We did publish the final toxin characterization in Science, and I'll show you some of the data and figures from this journal article. I guess it turns out not everybody reads Science. I think we need some more help with communication, so your group is going to be great. We documented it in a number of different bird species initially, and this work was done at the University of Georgia. The lead researcher was John Fisher at the Southeastern Cooperative Wildlife Disease Study. We call it SQUIDIS. I'll refer to them again, but they have done a lot of the initial work on characterizing the pathology. So we decided we needed to characterize what was going on in the reservoirs themselves. So we went out and said, what's unique about all of these reservoirs where the eagles have these brain lesions? Well, all of the locations had hydrilla. So that was kind of a red flag right there. But we hadn't really described any cyanobacteria that were particularly associated with hydrilla until we described this one. So this eagle killer really likes to live on hydrilla and other non-native plants, but it can grow on native plants too, especially if they co-occur with some of these invasives that get really dense. So when you're looking at it out in the field, you just see these little spots. It only grows on the underside of the leaf because, curiously, hydrilla actually has a pH differential from the bottom of the leaf to the top of the leaf, and it likes a very acid environment. That's what happens on the bottom of the leaf. So what we have is a very strange cyanobacteria that grows cryptically right on the leaf. And so the best way to see it is under this epifluorescent illumination, where you can knock out all the chlorophylls and just see the phycocyanins, or the blue-green algae pigments. And we know blue-green algae are the algae that are in freshwater that tend to produce dangerous toxins. So there was a hint that it could be a cyanobacteria to start with. These are kind of fun. This is an easy way to see it. If you were, say, in a field location, you can actually see these sparkling dots under that illumination, and it's covering the whole hydrilla leaf when it's really abundant. So this is kind of a lot, but we have tested a lot of different taxa, and we have only found that in the green tree frog, they don't seem to be particularly sensitive, but basically every other taxa we have tested has been sensitive, or we have found animals from the field that are suffering similar signs and have atoxin poisoning. So we continue to work on expanding our understanding of what it could be doing in these aquatic food chains, and how is it moving through them. But we're pretty sure it affects all of the taxa. And our most recent data are from some python tails that Mariah sent us from Florida. I guess those were part of the roundup, that she had some additional tails she could have shipped up. So we found, of the 10 that were sent to us, 5 of them are positive. So we're going to be working on this more, because obviously y'all know that the pythons are eating everything in the Everglades. Not everything, but they're eating a lot of the small mammals. And hydrilla is growing in lots of places across the US, and just about lots of places in Florida. And that expansion is co-occurring with the autochthonous hydrilla. So we think it's probably the most dangerous in terms of causing an expansion of this disease. But as I've said, this cyanobacteria can grow on other things. We started working in Florida on a suggestion from a biologist who said, hey, what if those snail kites are eating the new invasive apple snails, and they eat a lot of hydrilla? Well, he was right. It turns out that if you feed those apple snails to a chicken, the ones that were collected from Florida did cause vacuolar myelinopathy in the chickens. Now, we're not going to test any on the snail kites, but we would like to get more of these invasive apple snails to test them for the toxin, and to see in what part of the apple snail they're concentrated, because the snail kites can pick out some of, perhaps they take out the GI tract, and maybe they wouldn't be affected, but we need to know how toxic those apple snails are for them. One of my former students, Wes Guerin, developed a max scent model, which was based, of course, on the positive locations, which shows you in red where we would expect to be able to find it. And we actually had a lot more positive locations in Louisiana this year, so this has expanded quite a bit. And we had more, another positive location in Florida, in northern Florida, so that will expand. And I will be working on a new model for the new locations. Some of the newest locations we found during our our USGS Aquatic Invasive Species Project was, we're here in Louisiana. This is probably hard to see. This is when I make a map. When my students make a map, they're beautiful. When I make a map, they're this, yeah. So we have a number of new locations here in Louisiana, here in the Mississippi Flyway, pretty critical duck area. And this part of this work was enabled by some genetic detection that I'll tell you a little bit more about. So the toxin itself, we were able to characterize by using bioassays, and the zebrafish is one of the most widely used for testing an unknown toxin just to see, relatively speaking, how toxic is it. So if they, once they're exposed to the toxin, they begin to have some of the same symptoms that we saw in the birds. They can't right themselves. He's just flipping his little pectoral fins, can't move himself around. So just by visualizing the zebrafish, you can see when they're behaving normally and when they're not behaving normally. But she also quantified, Tabby Hortenstein quantified this behavior by looking at how much they move around, and it is kind of surprising. They're basically sitting still once they get toxin. And we've collaborated with Timo Niedermeyer in Germany, who is an excellent natural products chemist and has some very fancy machines which allow him to not only characterize the toxin, but he can visualize it on a hydrilla leaf. And this slide shows you the hydrilla leaf in the background and then our fluorescing cyanobacterial colonies. And that specifically is where he found this brominated toxin. So it's a pentabrominated biindole alkaloid, and you can see the characteristic peaks there. And we found that what it needed, what our cyanobacteria needed to make that toxin was bromide. And there's plenty of it in these environments, like Okeechobee has plenty, but we have not found a toxinose in Okeechobee yet. But in our Strom-Thurmond Reservoir, where we've lost lots of eagles, that has a higher, the highest bromine is in the hydrilla itself. It hyperaccumulates bromine, so it's a great place for cyanobacteria that needs the bromine in order to make its toxin molecule to live. We also found it in the sediment and just a trace amount in the water. So, you know, just like you think of with diquat dibromide, that's a pesticide, or herbicide, I'm sorry, it will stick to sediment very quickly. So it is a biindole alkaloid, and it has these 5 bromines attached, as well as a nitrile and an amine group. So we think that this side of the molecule is the most toxic portion, and that there may be derivatives besides the parent molecule that we have to look out for in the future. But the chickens that were gavaged with the pure toxin that we were able to synthesize are having the same vacuolar myelinopathy that the birds in the field exhibit. And we found that the crude extract, which includes sort of all the things you could extract from the cyanobacteria and hydrilla, is actually more intense than the pure toxin alone. And I'll talk a little bit about that, but it looks like our cyanobacteria makes quite a few other toxins, at least 10 others. And with electron microscopy, you can say specifically that this is the same type of vacuolar myelinopathy that the birds had, because you can look at these specific areas under the electron microscopy. You see oligodendrocytes are busting apart. That's very classic for the presentation of this in histology. And we found the toxin in the game fish in Thurmond Reservoir. Again, that's the site where so many of the eagles are dying. And the highest concentration of the toxin is still, we still find it in hydrilla. Like in terms of the total, how concentrated that toxin is, hydrilla has the highest. but we've also found it in bluegill, red ear, largemouth, and warmouth. And I'm showing you student data now, and I don't want anybody to do anything with this data yet until we have it out in publication form. So I'm very grateful that I can share the draft sort of the information with you all. But this one is from the science paper. We did find the toxin in the coot breast tissue as well as in the thigh tissue. So at this point, we know that toxin is neurotoxic. It's lipophilic, not water soluble, but it's soluble in fat. So that has a much higher risk for trophic transfer. And we have confirmed that. And we're seeing that there's an accumulation of the toxin enough in wild fish and game that we have concerns for people eating the fish and waterfowl from locations where this is happening. And this is a paper that came out in PNAS last year describing the highly toxic dolastatin derivatives that the cyanobacteria also makes. And this is a beautiful picture of the cyanobacteria under regular light with its culture. So if anyone is interested in looking into some of the other toxins that Autochthonous hydrocola makes, this paper will be available. And I've had a number of students who have been working over the past 5 to 7 years on this that I'm going to share with you. This is Ryan Henninger, and she did some videos to try to explain this to children and people who weren't scientists by using the animals, and they really respond to that. Also, if you have a cute little animal you're holding, then it makes you less nervous to talk. So I think it's kind of a good technique. We're also working on genetic detection. This is a paper by Linka Stenklova, a colleague in Czech Republic, who designed the PCR primers for Autochthonous. So we can say definitively that that is the same species in all those locations, or not in the case of. And this is Erica Klar, my master's student that finished in December, and she developed a LAMP assay for it, which is sort of a fancy field applicable PCR strategy. And it allows you to take this small isothermal amplification device out in the field, and you can target autochthon gene clusters so that you know whether or not the gene clusters to make the toxin are present, but it's also targeting the cyanobacterial species. So I think this will be a really good technique for more folks to use that are doing genetic detection work, because that is something that's field portable. And we were doing sort of a long, tedious extraction process. Yes, mortar and pestle starting it off, going all the way through to the LCMS MS, but we now have some better methods that Seth McWhorter, who's completing his PhD this summer, has developed using Quetcher's sample preparation, which means it's quick, easy, cheap, effective, rugged, safe. Those all sound good, right? So the process of getting an extract to test for a toxin is greatly, and it's faster and it's safer for the students to be processing. And we can do it with a MALDI as well as the HPLC MSMS. It seems to be not quite as sensitive, but we can certainly see the same peaks when we use it to look at the toxin in a matrix. All right, I'm going to talk a little bit about mammals as I'm finishing up here. I don't want to take up all your time in the meeting. We did find this beaver out at J. Strom Thurmond Reservoir. It was alive. It was very sick. Its gut was full of hydrilla, and it was having a hard time walking around. So once it was euthanized, we saw that we had hydrilla and autochthonous inside of the beaver. And the pathologist over at Squidus have reviewed these slides recently in light of more of our mammal being positive for autochtonotoxin and said, yes, we are definitely seeing white matter vacuolization. This is very similar to what we see in birds when they have a high degree of white matter vacuoles. Unfortunately, we found something similar. The Florida panthers, who also are having trouble walking, hind limb ataxia, and seem to have similar clinical signs that we've seen in the birds. And there's a lot of testing that has been done on these panthers and bobcats to understand what this new feline leukomyelinopathy, what's the cause of this disease. At this point, we can definitely say we're finding autochtonotoxin in panthers and bobcats from Southwest Florida. We can't say that's the cause of their death, but cats in general seem to be quite sensitive to brominated compounds. This is another one of Dr. Wilde's maps, so it's not very good, but this shows you the location of our positives from the panthers. This is what our highest one was way down here by Copeland. And these are the snake tails that were sent to us. Many of those were from similar locations, but I said, I got half of them were detects for the autochtonotoxin. So yeah, we had 20 and 10 were positive. I told you that less than that. And these are places where we know we've had hydrilla down there for a long time in the panthers habitat, and we've got apple snails in there now and pythons. So I think that I really would like to look at some new samples from this location, but I'd also like to look at some old samples, because with the fluorescence, we might be able to see it if it's on herbarium hydrilla. We might be able to see the cyanobacterial pigments, or we might be able to recover DNA from it. I've had some folks that at Georgia that have done that from herbarium specimens before. So just a thought, if there's access to some of those, we should try to look and see how far back we can document this in Florida. And this is Cassidy Brown, who's been working on wood ducks. They are failing in the same lakes, and we found toxin in the eggs, and we're working to see how long that persists within a system once you get rid of hydrilla. And Tobias Hames has completed a mouse trial, and he's the one who's been working on the Florida panthers. And Jeff Cullen has been working on looking at the autochtonotoxin in fish and isotopes and trying to compare their trophic status with their autochtonotoxin poisoning level. And as you can see, these are coming up very soon. So these are defenses that are within the next 2 weeks. And I'm going to show you a little bit of data here. This is, again, this preliminary data. So we'll have to verify everything, double verify everything before we publish. But we've had some quite high numbers, concentrations within a lot of the water, a lot of our fish and wildlife, but we have never found one higher than what the concentration is in hydrilla. So I'm pretty firm believer that we try to get rid of hydrilla when we find autochthonous growing on it, because it's a source of a toxin that is very persistent, lipid soluble, and it's moving through these aquatic ecosystems. So I think, maybe not very many people are going to eat coots, but people certainly eat some mallards and sunfish. And we're just recently asking more questions about, well, okay, so all this toxin is produced. Where's it going? It's in fish, it's in waterfowl, it's in sediment in the winter when you collect it, not real high, but you can still detect it just in the dirt that remains there. So the laboratory mouse trial was a way to test whether or not mammals were susceptible. And we did find that all of the mice that received the toxin gavage did develop lesions, and we had fairly high levels of toxin in the bodies of the mice following that exposure. So maybe I'll just leave this slide up and see if there's questions at this point. I don't want to take up all your time for the meeting.
Well, this is our feature presentation, so
Okay.
I'll give you all the time that you need to share the information. We don't have any questions in the chat at the moment. Is there anyone on the call with a question? Well, I'll start. I really like that idea of testing herbarium samples to kind of look at a timeline for the cyanobacteria through Florida. I have a good contact with UF's herbarium. I can put you in touch if you'd like to talk with them about some collaborative research.
What? I saw that Ian Finkston was on the call, and he's our collaborator too. I just wanted to say thanks to him for all of his help.
So we have a question now. Do we know all the species this affects, or is it still expanding?
No, I'm sure we're still expanding. I feel like we just sort of scratched the surface. We're just learning how to do this analysis in a way that is efficient enough to get samples processed and because animals die all the time. There's a lot of animals coming into Squidus. So we're investigating those that have unknown cause of death to see if the toxin is present. And that's where we found the positives for the eagles in Southwest Florida having a toxin. It's not a very high concentration, but I wouldn't have necessarily expected to find it prior to finding it in the panthers and then in the pythons.
Thank you. Thinking about the managers out there in areas where this is known or not known, what are some steps that people can do to help? Should we just make people aware of the hazards of eating wild caught food from these areas? Are there certain reporting that we can do if we see an animal with this type of behavior?
Yes. So if you do see a sick animal, in fact, not yesterday, 2 days ago, FWC called to say they had a sick coot. What were they to do with it? And so Katie Callahan, who's on this call too, she's my research professional, sent all the information to him to explain how he would contact Squidus, make sure they're expecting the bird, but then you got to ship it up there really fast so that they can do the analysis. And then they save tissues for us to test for a toxin. But I think the thing that this group might be able to help me more with is what are the other parts of the food chain that we need to be sampling? Like, I think we need to be sampling hydrilla and invasive apple snails. It'd be great if we could get alligators or alligator eggs or pythons, more of those and more eggs of pythons, because I know the bobcats are like sneaking in and eating them sometimes. There's some strange things when the predators are eating each other, right?
Yeah, that's a great question. Anyone in the group have any thoughts on what other species we should be monitoring for this?
Hey, this is Jake there at South Florida Water Management District. Potentially grass carp,
Yes.
common biocontrol.
Those things are hard to catch once you get them into a system, but absolutely grass carp, because it's the shorter the food chain, the more likely it makes it up to the top. I think that's why hydrilla and the coots are really pretty high levels. And then some of the herbivorous fish are going to be pretty high.
What about other more upland snakes? Do you think those could potentially be getting contaminated for their food chain, or is this more just around aquatic areas?
Well, we did test water snakes and Nerodia sipidon juveniles that we just collected from the field and fed them fish and tadpoles that had been feeding on hydrilla directly. And and they developed vacuolar myelinopathy. So I think it's more likely in an aquatic associated snake.
We had another question in the chat. The toxic and diseased animals from this toxin are primarily found where hydrilla is, correct?
Yes. We have, there are a couple of wildlife management areas in Georgia that have naiad infestations that are quite toxic also, that they're also quite dense infestations. So I think that's part of the secret, is that it's how densely is this vegetation growing? Does it like sort of take over the water column?
Sense. Any other questions from the group? Well, I will share with my monthly digest out to our SISMA listserv this information about the disease study. And if you have any other information that you would like for me to share out for them to get back to you, just let me know and I'll include that. But really appreciate your time and expertise. This is a great talk. And I'll go ahead and switch over to our SISMA updates.
Thank you.
Can you all see my screen okay? All right, so as far as upcoming events, we've got a lot of great things going on statewide. It's still relatively decent weather throughout most of the state, so we're getting a lot of cool work days that are going on, as well as outreach events across central and southern Florida. I want to point out Heartland SISMA's annual invasive workshop is middle of next month in April. They have 2 days, I believe, repeating the same content so that more people can attend. That is available through Eventbrite if you're interested in signing up for that. Looking out a little bit farther on. Into later on, again, more meetings and work days. North Central Florida SISMA is having a joint event with the Florida Native Plant Society at their annual conference here in Gainesville. We have virtual Python patrol, and then towards the end of April, we have a lot of really great kind of public, not really outreach, but public involvement events. So there's 3 different kind of invasive roundups going on in South Florida. So we have the removal of the swamp invaders exotic species fishing tournament at the Florida Lion Keys Fish Derby, and Southwest Florida SISMA's annual invasive freshwater fish roundup. So lots of great events if you're interested in getting some on the ground impacts out there down in South Florida. As far as announcements going on, FISC is starting the strategic planning process to update our goals and look where we're going to move in the future. And we'd really like your feedback. I'm going to throw the link to this in the chat so you can access it that way too. This is a short survey. Whether you're a member of FISC already or not, we'd be interested in hearing what kind of things you are looking for from our statewide organization. Also, the UF assessment is updating some of their assessments on different non-native plants. And so there's about 10 or so species that they're evaluating at the moment. And if you have any information on that, please email Dr. Sockman Kim, and he'll provide you more information on how to participate and provide that critical feedback for the assessment. Looking at the rest of our 2025 SISMA call schedule, great lineup this year. Next month, we'll be talking about the Inhabit tool, which looks at climate change and potential habitat, data-driven analysis for many different species across the country that can be used to help determine what potential EDR species, where other invasives might move into your area, how much suitable habitat you have for different invasive species on the landscape. And then in May, we'll be looking about the invasive snake lungworm parasite, another interesting impact to our native wildlife here in Florida. So this kind of wraps up our call. We had a quick meeting today, and hope everyone had a good time, learned a lot. I really appreciate Dr. Wilde and bringing an update on this really important disease that's affecting our wildlife. And if you have any questions, don't hesitate to reach out to her. But thank you everyone for your time. Appreciate it. Hope you have a great day.